I'm really happy to introduce our speaker tonight. Dr. Brian Hedlund holds a bachelor's degree in biology from the University of Illinois, a doctorate in microbiology from the University of Washington, and was a postdoctoral fellow at the University of Regensburg, Germany. Dr. Hedlund was hired at UNLV in 2003 and was recently promoted to Distinguished Professor in the School of Life Sciences. He has published over 160 scientific publications and has been a principal investigator on more than $9 million in extramural grants. Dr. Hedlund is the highest ranked author in Google Scholar with the keyword thermophiles and the fourth highest active researcher with the key term microbial systemics. That's right, systematics. He has also taught more than 2,600 students at UNLV, mainly in general microbiology, microbial ecology, graduate seminars, and scientific writing. His students frequently cite his enthusiasm, discussion of research and personal experiences, and overall dedication and support from his students, for his students. Please welcome Dr. Brian Hedlund.
Take this off because I can't stand still. So my shirt is my first prop. Has anybody been to Gerlach before? Anybody had the ravioli? Okay, you should go to Gerlach and try the ravioli. So where the pavement ends and where the thermophiles begin. Thermophiles are heat lovers. So I study microbes. I do a lot of different things, but my best stuff is studying microbes that love heat, thermophiles. And the best place to do that is in Gerlach. I really think so, partly because the landowners, David Jameson and his family, have just been so gracious to me and many of my friends and collaborators. And so he allows us to do things that would take lots and lots of paperwork and permitting and things that my friends, I don't study microbes in Yellowstone that much anymore because I can, because I have Dave and I can call him and I can ask him, can I do this? And he says, yes. And so we go up and do it. And so in a lot of ways we can, there's great research in Yellowstone too, but we can lead some of the things here in Nevada, but partly because of Dave and his family, and we can zoom through, whereas we don't get caught in the bureaucracy too much. So it's an incredibly lucky situation. So thank you.
Yeah.
Okay. Let's see. I'm going to start with a video. So that was done by a genomics company called Illumina, but they did such a good job, and I thought some of the visuals and things there were probably better than I could come up with, saying, imagine the hot spring looks like this, if you have a video that shows that. So I wanted to start there. So this wasn't a video, but it's important when thinking about hot spring biology to think about where the water comes from and where the chemistry comes from, because the chemistry is what feeds the microbes. And so, as was stated in the video, we have rain and snow, and that filters into the subsurface, and that flows in the subsurface for some amount of time. And in Nevada, we have a thinning, right? Because Nevada is getting torn apart. It's undergoing tectonic extension. And so the heated crust is moving upward. There's a shallow crust here. And so the water gets heated and it moves up. And one of the amazing things is that sometimes the water boils in the subsurface. And that's really cool because the chemistry from the liquid goes off one direction, and then the chemistry from the gas goes another direction. And so you get totally different springs. So some of the springs would become really sulfurous. I brought some sulfur and some sulfide that I can pass around. If you're brave, you can smell the sulfide, but don't take too much of a good breath of it. But the microbes, some of them love to eat this stuff. So we'll talk about that a little bit because it's kind of crazy. So that would go in the gas phase, and then the water would be stripped of the gases. And so you get really different microbes. And in fact, these two different kinds of springs are buffered by different chemistry. So this one is buffered by carbon dioxide and bicarbonate, so carbon dioxide from the atmosphere and bicarbonate from the water. And this one is actually buffered by sulfuric acid. The microbes make sulfuric acid by oxidizing those sulfur compounds, and so they're usually around pH 2. So not only they're hot, but they're hot acid. So sometimes doing research, those kinds of springs are much more common in Yellowstone and in some parts of China that I work at, and they actually eat away at people's clothes. So sometimes we get, people's clothes degrading in the middle of the research. One of the interesting things we found pretty early on studying Great Boiling Spring is that there's a really strong quantitative relationship between the temperature of the spring, of the sample we take, and in this case we're just taking samples from different parts of the spring. So the chemistry is more or less the same, we're only changing the temperature. And then this is operational taxonomic units, so you could ignore that, but basically this green line is the number of species, and then the number of genera and families and classes and phyla. And so, you can see these lines are cranking down pretty hard, and at the hottest sources of Great Boiling Spring, we've only got something like 20, 30 species in there, that's it. And these are pretty exhaustively studied. We've, through the years, sequenced probably millions of DNA sequences from the spring, and yeah, we get 20 or 30, that's it. And so it makes it an interesting environment because it's so simple. It'd be very different from a soil where you might have 5,000 species of bacteria in a teaspoon of soil, something like that. So what are these things? You don't have many species, but what the heck are they? Also in the video, interesting thing is the upper temperature limit of photosynthesis. So that means that the energetics of these springs, the bioenergetics, is really different. And so being in Fallon, of course, we have a lot of agriculture and everything with it. The cows eat the alfalfa and so on. But the carbon and the energy for life in most ecosystems is really from sunlight. Okay? Here, it's too hot, and you can see very easily that it's too hot. So you can see this 73 degrees right here, okay? And up here, it looks like there's no life, but there is, I promise. And here's just a picture of an outflow channel where you can see to the left is hot and you're changing, getting cool. So this is something that's fun for us to study, and also for us to think about how life works, where photosynthesis isn't directly providing the carbon and the energy, because it's pretty different. Okay. If you look in the main water, the big pool of Great Boiling Spring, there are two species that are more than 99.99%. It's amazing. You don't find this in nature, okay? This is really weird. Here we stained them with different colors. Dave, you get pink. Is that okay? Okay, so the pink one is called, we named it Thermocrinus jamesonii, and it means Dave Jameson's, that's referring to him, and then hot hair, because thermo is hot, and crinus refers to hair in Latin. And so we thought it was kind of cute. I don't know if you like it. The other one is called a pyrobaculum, and my colleague Jeremy Dodsworth is naming that one. So we don't have a name yet for that one, but the genus pyrobaculum means a fire stick. So it's always fun naming microbes. Okay. So I'm going to tell a little bit of a story about two different microbes that kind of show, maybe allow me to talk about some of the stuff we do. And the first one is Thermochrinus jamesonii. So I'll tell you a little bit about the history. So the history of that one was a little bit easy for us because we knew from DNA sequencing that there were only two main microbes there. And my where I did my postdoctoral work in Germany had discovered this thing called Thermochrinus ruber, hot red hair. And we could see that this was related to Thermochrinus ruber. And so I took out my recipes, my recipe book on how to grow Thermochrinus ruber, and we grew Thermochrinus jamesonii. I say it's easy, but there's only this phylum of bacteria. There's probably like certainly less than 10 labs in the world that would grow them. They're not that easy, but we could grow them in liquid. So we grow them in bottles like this because these are sealed, and we can control the gas, and we have to control the gas because they're very picky about different gases. A lot of them don't like oxygen at all. Thermochrinus actually does, but we have to control how much. So I can pass this around. That's actually a really old culture of archaea right there. But yeah, they grew up in liquid, and we could see them growing up. You know, it becomes cloudy, like a Hefeweizen or a Weissbier. It becomes cloudy. We could see it. Not that thick, but you know, you can see it. And we plated on auger plates. And because it's high temperature, we use this thing that I call a bomb. So this is my own design. It's a can for spraying paint, and then I modified all the fittings on top to withstand high temperature and pressure with some automotive parts and stuff like that. But this is a quick connect, and this is a little septum, so we can put whatever gases we want in there. And so this is, you can open it if you want. But we put petri dishes in there, and I think most people have maybe seen petri dishes. It is a little stinky, but you can see microbes growing on there. That's actually E. coli, so that's something from your intestine, and it's dead, so it's pretty safe. You can eat it if you're hungry. But anyway, so Thermochrinus didn't make colonies this big. This isn't actually Thermochrinus, but it made little tiny colonies, and we could pick them. These colonies arise because a single cell fell on, we put it onto to the medium, and it liked what it was eating there, and it grew up. A single cell grew up. That might be like, what do you think, 10 million, 100 million cells or something like that. The Thermochrinus ones are smaller, so it's not that many cells, but we can pick that one. And if we keep passing it on the auger, we can be sure that it's a pure organism. It's what we call a pure culture. And the history of microbiology is a lot focused on pure cultures because it's easy to study things that are pure. It's hard to study things that are mixes. It's mentioned in the video. Okay, so when you purify the organism, you look in the microscope and you know what you're looking at. And so this is not pink colored, but this is Thermochrinus jamesonii as a pure culture. And this is an electron micrograph of what that organism looks like. And since we had it pure, it's pretty easy to do experiments because when I go in the lab, I control the chemistry that goes into this, right? It's just like cooking, right? So I decide, well, maybe it likes to eat this, and I can add that, and then I have another petri dish or another bottle that doesn't have it, and I can see if it likes it, right? And so this is pretty simple stuff, actually. It's with the same thing I do for my dog. Do you like this treat? You don't like it? Yeah, I mean, it's not that hard. And so by doing that and studying it in the lab, we found that it's hyperthermophilic, so it grows between 70 and 85 Celsius. Its optimal growth temperature is 80, so it's pretty high. There's a few bacteria that can grow higher than that, but this is up pretty high. Some of the interesting stuff, I'm passing around sulfur, the sulfur flowers. That's the yellow the yellow in there. They eat sulfur, and so I brought an apple here, and I eat this apple, and it has sugars and some other things. And I breathe and I oxidize the sugars with the oxygen that I'm breathing in. Okay, and obviously I get nutrients from that apple and everything too. Sulfur is really interesting. I mean, all they're getting is energy. That they're not getting organic carbon from that to build or anything. That's just energy. So that's a little bit of a different thing. People aren't really used to the idea that microbes can eat a mineral, but it's totally normal. It's totally fine in microbiology. They can also breathe minerals too. Here I brought a couple of bottles of rusty water. All right, this is hematite up on top. This is like red rock near Las Vegas. And this is goethite, so it's rust. This is just rusty water, right? And this is a bottle where I grew some archaea that instead of breathing oxygen, they're breathing this goethite, and they made magnetite. And so you can see this is all hung up on the magnet here. So I'll pass this around. Microbes are pretty cool. Thermocritus can't do that, but but I wanted to at least introduce that. It also oxidizes hydrogen gas. Okay, I've got a slide to talk about hydrogen gas, or a couple slides. And you might be wondering if it's oxidizing, it's getting energy by oxidizing hydrogen gas or sulfur powder. Where does it get carbon from? And it gets carbon from the same place that plants do. It just breathes it in, and it's very expensive to do that. But plants, there's one biochemical pathway for plants to use carbon dioxide. There's a new one discovered last year. There's at least 7 that bacteria and archaea, different biochemical pathways to use CO2 for anabolic purposes, to build the cell. So Thermocritus jamesonii can do that. It can also eat some organic carbon too, some proteins and stuff. But it's totally fine using carbon dioxide. So I sometimes say that David Jameson is such a great guy that he can walk on water. All right. So I mentioned a little bit about Thermocritus jamesonii eating sulfur. It uses a couple different forms of sulfur, and this is the elemental form. That's the yellow powder I'm passing around. That's brimstone from the Bible. That's what that is. Okay. And hydrogen gas. This is H2. And what they're doing is just shuttling electrons from these things to oxygen. And it's not that different from a human. We shuttle electrons from pizza or apples onto oxygen. It's an exergonic reaction. It yields free energy. And the same thing for our car. We put fossil fuels in, shuttle the electrons from the gasoline to the oxygen, yields free energy. So it's not that crazy. It sounds weird, but it's not that crazy. Anybody know what this is? Yeah, it's a Hindenburg. So the Hindenburg has the same energetics as Thermocritus jamesonii. It takes hydrogen gas, 1 molecule of hydrogen gas, one of the atoms of oxygen from molecular oxygen O2 that we're breathing all the time, and it combines those and makes water, and that gives a lot of energy, right? And so if the Hindenburg can explode on that, certainly microbes can grow on that. It's a good thing to eat. It's actually a great thing to eat. And I don't want to go into the biochemistry too much, but it's pretty simple. They have an enzyme called a hydrogenase, and the hydrogenase splits hydrogen, H2, into electrons and protons. This is acid. It kicks the acid out of the cell, and it shuttles the electrons through electron carriers in the membrane. This is actually a lot like a wire, like electrons flow down a wire. Some of these things are metals, and the electrons just flowing along just like a wire. And then the electrons eventually go on to reduce oxygen to make water. That's the end of the reaction. And then this is basically like a battery, so that's acidic outside and it's not acidic inside. And when the acid comes back in, the cells can make ATP, and that's the energy currency that they use. It's like money. So biochemical money. All right, so these groups, these microbes, the group is called Aquafacoda, the water makers, because they have this reaction, they make water. That's what it's a pretty clean reaction. So this is why hydrogen is thought of in some ways as being a good alternative fuel, because it's pretty clean. Okay. Thermochrinus jamesonii doesn't, Thermochrinus doesn't only live in Nevada. There are friends that live different places. This is a panel that I put together from Octopus Spring in Yellowstone National Park. This was the original Thermochrinus that was discovered, Thermochrinus ruber, hot red hair. It looks like red hair. You can sometimes see hair-like stuff at GBS if you look around. I think that's Thermochrinus jamesonii, but I'm not totally sure. But this is massive amounts, and if you look in, it's Thermochrinus. This is a paper I'm trying to get published right now in China. And we found that actually it actually looks like Thermochrinus albus, hot white hair, but it's not. It's related, but it's not the same genus. So we're trying to name that organism, but it has a similar metabolism and it looks somewhat similar. So it's what we call Pampini fervens sericum. Okay, but it's a close relative. It's a close relative, same family. Okay. All right, so we talked about one of the major microbes in the spring water, Thermochrinus jamesonii, and I just wanted to talk about, I would say, the dominant bacterium in the spring water is Thermochrinus jamesonii, and I wanted to tell you about the dominant bacterium in the sediments. So Thermochrinus jamesonii is really abundant here. It's not very abundant in the sediments. If you look in the sediments, this is based on DNA analyses. The most abundant organism, and it depends sometimes when we go there. So the spring changes, it makes it confusing for us. But the most abundant organism is something we call Fervidobacter saccharide. And there we've stained for it. It's not a great picture because it's hard to deal with the mud, right? It's easier to image things in the water. In the mud, there's all this mud in the way. There's minerals and stuff in the way. Okay, but we can do it. And Thermochrinus jamesonii they're not easy to study by any means, but people have studied the genus Thermochrinus before. 2 different species had been studied. Thermochrinus jamesonii is the 3rd. Okay. Fervidobacter saccharide is off in space, evolutionary space, no close relatives. Okay. And I wanted to talk about it a little bit to introduce this idea. Sometimes it's called the uncultivated majority, meaning that the majority of microbes out in nature haven't been grown, cultivated in a laboratory or studied. Okay? Sometimes we call it microbial dark matter, so like dark matter in astrophysics, except that it's microbial dark matter. So we can find it because we can see its DNA and we can see its proteins and things like that. But when we try to grow it and study it, it just says we're not really interested. We don't care what you think. But in this case, we were actually smart enough to win. Thank you. So anyway, I call these the dark branches on the tree of life, and this is actually the branch that Fervidobacter saccharide comes from. Before we gave it a name, it was called OCT-SPA1-106. So we think Fervidobacter is a better name. All right. Anyway, this is just some of the numbers, but this just says that of the major lineages, phyla are over 1 billion years old. So if we count the number of lineages of bacteria and archaea that are over 1 billion years old, we've only grown in the lab about 1/4 or a little bit less than 1/4 of those. So the others are out there and they're there, but we don't really know how to study them very well. We're getting better, but okay. So one of the things that we've been doing with friends, this isn't my lab, but it's my friends, so it's always good to be good to friends. So I have a few different labs. One is the Bigelow Ocean Science Lab in Maine. Another, Stanford a little bit, but also the Joint Genome Institute in Berkeley, California. Thank you. And so these are a couple of the only labs in the world that are specialized in clean sorts of single cells of microbes. And this is a chip from Stanford. We were really early doing this, and this is the old way. The new way is easier. But in this case, what we would do is, this is a little microfluidic chip, it's sterile, and we would load a sample from Great Boiling Spring or some microbes that we were growing from Great Boiling Spring, but still a mixture. And we would load them into this, and we would use a laser. People know what a tractor beam is from Star Trek? So this is basically a tractor beam, but tractor beams, you have to get a really strong laser to get that to work. But this is a tractor beam for microbes. And so we put an infrared, it's like a harmless wavelength, onto the cell. And the beam, actually, the forces of the light diffracting through the cytoplasm, which is dense, causes the cell to remain in the center. And then we can move the laser beam around, and the cell sticks in the center. And so we can separate single cells from all the other cells. So instead of purifying them the traditional way, which would be this, we're purifying them with a laser, with a tractor beam. And so once we do that, we could try to grow them, but actually it's pretty rare to do that. I think me and my friends are the only people in the world doing that now. But what is done, especially by Bigelow and the Joint Genome Institute, is single-cell genomics. And so the single cells are separated from the pack. They're broken open. That's actually the hardest step, is to break them open. They don't break open easily. Then we amplify the DNA, because there's about a femtogram, 10 to the minus 15 grams of DNA in a cell. It's a tiny, tiny amount. Because you're amplifying, you have to be super, super, super clean, because any contamination, you'll just amplify the contamination. So that's why only a couple labs in the world do this. Okay. Then we can sequence that DNA. We assemble the pieces into like a big fragments. We annotate it, which means predicting what kinds of genes are present. And then we interpret it. We think about, hey, okay, we got this genome from a cell, we got its DNA, and you know, based on what we know about different genes and how they function, what do we think the cell eats? What do we think the cell looks like? Who do we think the cell interacts with? Things like that, things we think about. And so I was part of the biggest, I guess the first large-scale single-cell genomics publication. I was like a middle author. I was lucky. And we had 200 single cells, and I was lucky that we had I think 7 or 8 sites over the world that were selected as like the first test cases, and Great Boiling Spring was one of those. And so we got single cell, we got genomes from the single cells. This is a version of the Tree of Life. And we got this thing, this OxSpa 106, whatever, I forgot the name. We got it. And we were then able to look into the DNA and think about what the heck this thing is, because otherwise we had no idea. We knew where we could get it, but that's pretty much, we knew it was bacteria, but we didn't really know much else. But once we got the DNA, we could look at the DNA and think about it, and the thing that jumped at us right away is that it has lots and lots of genes for eating sugar. And it's not as exciting as sulfur and stuff like that. But this is what we saw. You know, I didn't choose that. And by the way, just because it eats sugar doesn't mean that it doesn't do other cool things. So we know a little bit. There's a lot we don't know. But yeah, so we decided to call it, instead of the OxSpa 106 thing, we decided to call it Forvidibacter saccharoi. And so Forvidibacter basically means a hot rod, Forvidibacter hot rod. and saccharide refers to sugar. And so one of my students is really into AI images, and so he made this hot rod sugar eater with a fork. And his family also likes hot rods, so he made this to show his dad. So this is what we were imagining for Bifidobacter saccharide, but we still just had DNA at that point, and I don't know, like, we're kind of smart, but we're not that smart, you know? But it's kind of limited to have DNA, and the closest relative is over 1 billion years old, and how do I predict in 1 billion years of evolution what the heck this thing is, you know? So, but we really did have this prediction that it eats sugar. This one isn't published yet, by the way, the best stuff. So it has all these genes to break sugar-sugar bonds. So for a polysaccharide, it's made of many sugars, like starch or cellulose or things like that. And so it has these predicted enzymes that break those into smaller pieces and eventually into monosaccharides like glucose and sucrose and sugar. It's the sweet tooth, right? And so that's what we could see in the genome. And we compared the number of predicted glycoside hydrolases in the genome, and it's 117 different copies of those types of genes. It's way more than any other known thermophilic microbe. So the genome was screaming, I like sugar, I like sugar. And so what we did, Nancy was in the video, I told Nancy, look at these glycoside hydrolases and try to predict what kinds of sugars they're going to eat, what kinds of polysaccharides. And so Nancy's really smart, and so she did this, and she ended up predicting substrates. And so this is kind of confusing, but basically we had this list of food that we thought that it would like to eat based on the DNA that we got. And I'm not going to go through all of this, but we did all of this testing. So these are in these little culture bottles, and we just have, vary the recipe by one thing at a time, change the temperature, do this, do that. And we follow their growth either by staining them. I showed the staining, we can see them in the microscope. That's kind of tricky though. Or by looking at their DNA. And so in this case, this is just a bunch of different conditions. This is an array of conditions. And the light green is the Bifidobacter saccharide, and it's growing to pretty high abundances there. So I sometimes think about fishing, and like we had a fish on the hook, we have to get it in, but we had a fish on the hook. Okay. All right. So I told Nancy, get back to the lab and work more. and so she looked to see how much oxygen it likes. It likes a little bit of oxygen, like half as much as we like. Okay. We looked to see how fast it grows, and we were really lucky. It grew really fast because we knew how to feed. We knew that it liked sugars. And so it was growing really fast. The other microbes were growing kind of slow. That's good. And then we tested exactly which polysaccharides it likes. So I won't go through all the data, but we had a good idea of what they like to eat. And after a few months of just growing them in these liquid with no way of separating single cells, we had 99.6% Bifidobacter saccharide. We look in the microscope and it looks like Bifidobacter saccharide. It's not quite exactly pure yet, but it's almost there. So we tried to grow them on petri dishes on solid media, and they said, no, they don't like it. We tried all the tricks that I know. We even tried to grow them on silicate, like it's like solidified glass, which should be as inert as possible, and they didn't like it. So anyway, they didn't like it. Oops, I messed that up. What this is showing here, this is that single cell chip that I talked about, single cell genomics. And if you think about it, if we can separate single cells and break them open, we could also separate single cells and then throw the single cell in the bottle and see if it grows. And so we threw 10 single cells. It took us like 10 hours. It's actually like playing a video game, kind of. So like zap that one and grab this one. We picked 10 single cells into 10 different bottles. One grew. But you only need one, right? Sometimes it's good to be lucky. So one of them grew. And we had a pure organism. This is electron microscopy of this organism. It doesn't look that exciting. It's a beautiful image, but it's a bacterium. There's not a ton to see. It is hyperthermophilic. Its optimal temperature is about 83 to grow. So there's only a few bacteria that can grow higher than that temperature. There's a lot of information here, but the most important is up here. So here we're just testing different sugars. We have the pure organism now. So we can just ask it, do you like this? Do you like this? Do you like this? And so that first bar, this is a bar graph, has no carbon, so that they shouldn't, they can't grow without carbon. Nothing can grow without carbon, at least that I know of. And then we're just testing polysaccharides, different types of polysaccharides. This is plant biomass. The Department of Energy is interested for biofuels purposes. And then monosaccharides, so use glucose, right? And it can grow a little bit on protein, but not very well. It loves these sugars. So we were completely correct in this, sweet tooth prediction. These are data on gene expression, and I guess I can say that when they're eating sugars, they make these enzymes that we thought they would make to eat the sugars, and that's basically what that says. And the different enzymes are made when we give them different polysaccharides, which makes a whole lot of sense. So I have all these 117 different copies, and they make these enzymes depending on which polysaccharide they're eating or which sugar they're eating. Okay, that makes a lot of sense. Gosh, we did a lot of work with this. We worked with the lab again in Berkeley, the Joint Genome Institute, and we synthesized in the lab all 117 genes, and we put them into E. coli. E. coli is like a lab rat or something, and so we asked E. coli to make the pure enzyme, to make the enzyme, and then we could purify the enzymes from E. coli. So we purified 117. Actually, 10 didn't work, so we only 107. So we purified 107 proteins, and we tested them against, I think this is 16 different polysaccharides, and most of them didn't work. So this is what we call a heat map, and the more enzymatic activity makes it more red, and then the ones with stars are where we confirm the activity. So most everything failed, which is just the way it is sometimes. But we got 9. We got 9 out of 117. We might get some more. But 9 of them worked, and we can study the activities of those individual enzymes. And not surprisingly, they work. This one works optimally at 100 Celsius. That's boiling. And that's the optimal temperature for that enzyme. Well, of course, Forbidobacter saccharide loves heat, and so all these enzymes work at high temperature. This is a little complicated, but this is an evolutionary tree that we made with the genus Forbidobacter. I called my friends, and I said, I have friends all over the world, and I said, well, do you have any Forbidobacter genomes that we can borrow? So I called my friends in China, and my friends in Yellowstone, and my friends in New Zealand, and everywhere there's hot springs, right? And I said, give us some genomes, we'll add you to the team. So it's something I'd like to do. I don't mind having a big team because you can be powerful that way. And so we put together, these are species representatives. So we have more genomes, but these are just the one genome per unique species. And this is an evolutionary tree, and this group up here, well, this is the genus Forbidobacter. So you can see we named some of them. It's kind of interesting. We find them in Japan, in Canada. We find them in different places. We find them in Yellowstone. Okay, but our species only lives here. All right, but we find related species. This is the class. Things above this are the class Forbidobacteria. None of the others have been grown before. Down here, there are some organisms with names. A few of these have been grown before. This whole group is a family tree, phylogenetic tree for a phylum called Armatomonadota. And we made this tree, and lo and behold, we wanted to ask, we learned that Forbidobacter sacchari loves to eat sugars. Do they all love to eat sugars over 2 billion years of evolution? And the answer is yes. And so nobody knew this before. We're still trying to publish it, so people don't know it yet. But yeah, so this blue bar is a bar graph showing the number of predicted glycoside hydrolase genes to degrade polysaccharides. Forbidobacter sacchari is actually not that good. And so you look at some of these bars, they have 350 different copies instead of 117. And so we think now, taking the information we learned at Great Boiling Spring, we think we just discovered that this 2 billion year old lineage, that they all love to eat sugars. So we're trying to tell the world, hey, if you're interested in these, give them sugars. It's not going to be that hard, we think, of course. So there's more stuff here. This is showing genes for breathing air. Forbidobacter sacchari likes to breathe air. So you can see some of these blue boxes, which means they have genes to breathe air. But most of them don't like to breathe air. So there's, in microbiology, different microbes share genes with their friends. And so the genes for breathing oxygen are sort of shared back and forth between different microbes. But most of them are anaerobic. And so we would need to grow them with a controlled atmosphere to be able to grow them. All right. We're not really sure, there's some plants around. And, in the video we said there are plants around, and we've done some calculations and some looking at isotopes to try to estimate how much of the nitrogen or the hydrogen or the carbon is coming from plants versus the spring water coming up. The nitrogen's all from the spring water, but the carbon hydrogen's a little complicated. But anyway, there's not a ton of plant material. It looks like a swimming pool in there, right? And so we actually think that maybe Forbidobacter sacchari and maybe some of the other relatives, maybe they like microbial polysaccharide instead of plant polysaccharides. So that's something we're working on right now to try to understand like the real life of this organism. Okay. I forgot that the video had some viruses, so I'm more or less done, almost done. But I wanted to show some pictures of viruses, so the video had some. This is some of my postdoctoral work when I was in Germany. So this is from 2 springs in Yellowstone National Park, and just different viruses we could see. They're wild, so just different shapes and stuff. A couple of my collaborators at the Pasteur Institute in Paris have studied these their whole career, of studying these viruses. So I'd let them have that. I have other stuff to do, but they're really neat. And these are, they're not giving measles to the microbes. They're attacking the microbes in the spring, right? The cells, the bacteria and archaea. I wanted to just talk about, this talk is about Nevada Springs, and so I just wanted to sort of come back a little bit to Nevada Springs in a little bit more general way, and maybe just think about what I can say about the microbiology of Nevada Springs, because I guess I'm the expert. I've been studying them for 20 years, and I can't think of, there's nobody else anywhere close to me. So even though I know about this much, it's that much more than most other people. And so one question that I get a lot is whether the microbes in Nevada Springs or Great Boiling Spring are unique. Are they endemic? Are they special to these springs? And I have a couple of answers, and they both have question marks, so that's just where we are. But the first answer I give is no. And that no is for genera. So genera, we're the genus Homo, or my dog is Canis, right? So that's pretty closely related. And what we find is that it's nearly impossible to find a genus of bacteria or archaea that are only in one state or one area or even one continent. You saw that with Forbidobacter sacchari. It has relatives in Asia, all over the place, in Canada, in Yellowstone. And so it's pretty funky, but in a microbial genus might be something like 50 million years old, something like that. So they're passing between the continents within that time period. I think it's probably volcanism, like volcanoes and these things get launched, I think. So this is so special and rare that a high-profile paper just came out from some friends of mine studying a relative of Thermocrinus jamesonii, a different genus, but a closely related genus. They named Venena vibrio, and they think that genus only lives in New Zealand. And I might have some cases where I think I can see that in some of the data, but it's hard to find. It's really hard to find. So that you wouldn't, with plants or animals, you would see unique plants or animals in Nevada. So it's a little bit different. I think part of the problem is that we define our microbial taxonomy as a deeper time taxonomy, and so what we call a species or a genus isn't exactly the same. But they also spread easier. They just do spread easier. Okay. And then I have a yes answer. So I say that probably most species in Nevada hot springs are endemic, probably to individual geothermal areas. And I do have some data on this. I'm trying to get this published, haven't published it yet. As I've said before, for Vitibacter saccharoi, or I don't know if I said this, but it's only in Great Boiling Spring as far as we can tell. We can find other members of the genus, even in China and Japan, but the species, I don't, if we go around northern Nevada, we could see exactly where it lives. But as far as I know, that's the only place I've ever seen it, and I've tried pretty hard, okay? I also have some data from Cold Springs, and so this is, you'll notice a decent number, 23 springs, 9, 6, 9, 19. We've like about doubled the number of springs. So this is between Pahrump, Nevada and Bishop, California, sampling springs and that sort of transect. And so we've grouped them just by either the mountain range or the valley. And the vast majority, if you look, this is a weird Venn diagram, but most of basically the species are only found in one of these valleys or mountain ranges and not others. So only 453 that's found in all of these areas. This was surprising to me. So what we calculate is that 26 to 79%, if you go to a random cold spring of the species, I think are only in that spring or in the springs right around it. And we've sequenced something like 60 million DNA sequences now from these springs. And so we're trying pretty hard and spending money and trying. And we've tested this a few different ways, and so it looks like it's true. So I don't really understand it exactly, but it looks like what we see. And so I mentioned this partly because we do have this goal of 50% clean energy in Nevada by 2030, and there aren't that many hot springs around. They're special, right? They're special, and Great Boiling Spring and most of the hot springs are taking in ice age water. So this is unusual water. It's heated to unusual temperatures. There are long distances between them. And, geothermal is a big deal in northern Nevada. And so there is some pressure. And I guess my view would be that we should probably be a little careful. Just sort of thinking about, people may or may not care about microbes, but the geological setting there is very unique, right? And that's not coming back if we ruin it. The other thing is the crust is really thin up here, and so you don't really have to go to a spring to do this. So I just want people to think carefully about it. I don't have an answer exactly. So here I just wanted to thank some of the people from my lab group, formerly or present, who are really important. So Jeremy Dodsworth is my former postdoc, and he's a professor, he's a chair now at Cal State San Bernardino. Marika Palmer just left my lab. She started her own lab at the University of Manitoba in Winnipeg. And then these are a few. Nancy was in the video, but Deng-Shun Lai is a great student, did a lot of this work. Nancy and Jonathan Covington. I've had money from a number of places. I do try to work hard to put it to good use. So anyway, that's all I have.